It's a great pleasure today to try something rather new. We have an extraordinary talk by Professor Nanna MacAulay, who, despite the name, is fully Danish, although she has spent some time on other continents. She's a professor in University of Copenhagen, and she has some really interesting research. She would herself say that she's not a neuroscientist, but of course, what she does, interested in how things travel across membranes, is highly relevant to a lot of us. And specifically, of course, how it is that the water on the brain would change us. So she's, given that she's coming here to this crazy center, she's tried a new title for the first time in her life. She told me it's called, and I remember her coming up with the title. I just want to state that for the record. Water in the Brain, Challenges and Opportunities for Survival and Thriving. And I think you will come to see the wisdom of that title, this brilliant title of Nanna's, in the work. You will have seen her virtually, or at least somebody who looks like her moving on your screen at the moment. For now, it's a great pleasure to introduce in person. Nanna, please come and tell us about your exciting research.
Thank you very much, Morten, and thank you for the invitation to come here today. I've been very excited. I've never been to Oxford before, and I'm super thrilled and blown away, and I've never had so much coffee and so much chocolate in such a short time, and we're not about to finish, as I understand. And it was Morten that came up with this title. I just want to let you know. But it basically was because what I do is a little bit more molecular than what you guys normally do. So we wanted to expand it into sounding like a little bit bigger. But I have gotten rid of most of the molecular stuff, and I'm trying to make it so hopefully you'll be able to follow along the way. But the title that I normally have, I've actually added here at the bottom. Cerebrospinal fluid secretion, like the bumblebee that cannot fly. And we'll get back to the bumblebee a little bit later. So we normally actually don't do very much MRI scanning at all. It's something more recent that we're starting. So this is actually a pilot scan of my brain, because we're interested in how CSF can be measured non-invasively in humans. So we're interested in aqueductal CSF flow, but that will be for next time. I'm just super excited about seeing my own brain in a scan. You guys see this all the time, but I mean, this really gave me a major kick, or gave me pleasure, as Morten would call it. Anyway, so our brain is actually full of water. It contains 80% water, pretty much. And this water is replenished daily at a rate of approximately 500 milliliters that is secreted from our vasculature and into the brain. And generally, we're actually quite content with this amount of fluid. So some of it surrounds our brain and therefore serves as protection from mechanical insult. And it also resides in the ventricles, which you probably often see on your scans. And because the CSF in the ventricles is in continuum with the interstitial fluid surrounding our brains, it's really the only transport route for nutrient delivery, signal molecule dispersion, or waste clearance. And this is irrespective of whether one is into lymphatics or not. It is the only way to transfer things between neurons and glial cells and in and out of the brain. So generally, we're very happy with this amount of fluid, but in a lot of different pathologies, we have too much of this fluid in our brain. And it disperses differently depending on the underlying etiology. So you just see some of them here. So those that have enlarged ventricles, you see this enlarged black parts in the central part of the brain, those is what we call hydrocephalus. That means water on the brain. And you have probably seen some of these horrible children, pictures of small children where their whole head gets enlarged because they have soft skulls. So this expansion of the fluid would lead to this enlarged cavity. And this is not the case for us because our skulls are hard. So we will not expand our brains as such. So this type of pediatric hydrocephalus is actually quite common, as you see here, mainly in the third world countries. But different types of hydrocephalus can be prevalent in different age groups, as you'll see some suggestions of here. And the one called normal pressure hydrocephalus, Morten and I briefly talked about it after lunch, it actually may target as many as 2% of the elderly population and can actually be seen as a kind of a curable dementia. One can actually relieve the elevated pressure by removing the fluid.
I have that diagnosis.
You have normal pressure hydrocephalus?
Currently, yes.
Yeah, have you gotten a shunt or you're...
I don't know the details. I'm hoping to find out more about it.
Yes, so people actually do not know very much about normal pressure hydrocephalus and why it comes about. But many people get better by having some of the fluid drained from their brain. So they basically
I had that done.
Yes, so that might eventually, and that actually helps a lot of people, and we don't even know why. I suggested more to scan people that you before and after, so it would be a lovely suggestion for that. But taking all these hydrocephalus types all together, it is actually the most common entity addressed by neurosurgeons, and is also the most common childhood disorder, accounting for up to 3% of pediatric hospital charges. And it's not the water in itself that's damaging to these patients, it is elevated intracranial pressure that comes with it. So the intracranial pressure would be directly a function of the fluid content in the brain, which is the balance of the CSF being produced and the CSF outflow. So we could have a scenario with a reduced CSF drainage that will lead to a reduced CSF outflow and therefore an elevated brain fluid content. Alternatively, we could have an elevated rate of CSF production, which would also lead to an elevated brain fluid content. And these, either alone or in combination, would give us this elevated intracranial pressure that is highly damaging to the patient and even fatal if left untreated. And this has been a major clinical problem for a very long time. So this is actually a picture from the Danish National Museum where you see a cranium from the Stone Age, so 10,000 years ago, or 10,000 BC. And you probably notice the extra hole in the cranium up at top. And one could contemplate that this could be due to a hit on the head from the neighboring tribe, but this is actually not the case. This is an example of the earliest known surgical procedure, which is called trepanation. So here one makes a hole in the cranium to relieve the elevated intracranial pressure. And this is probably meant as a joke, but this is the only way they could have done it back in the Stone Age. So according to the text along the side of this picture, this patient actually survived and lived on now with this hole in the cranium. So it's a way to relieve an elevated intracranial pressure. And this was basically continued up through the Roman Empire, into the later medieval times, into the Renaissance, and also up through the 19th century, where in all cases, surgeons would relieve the elevated intracranial pressure by creating a hole in the skull. And this is actually, scary enough, more or less what we do today, now just slightly more refined and under anesthesia, of course. So the surgeons can place a shunt in the ventricles that basically deliver the excess fluid into the abdomen. And this relieves the elevated intracranial pressure, but these shunts only have a median lifetime of approximately 2 to 4 years, after which these patients have to undergo semi-acute neurosurgery, often for the rest of their lives. One could also make an extra hole in the floor of the third ventricle, endoscopic third ventriculostomy, to create another drainage route from CSF. Or one could even go as far as cauterize the cord plexus, which is the tissue that creates this fluid or secretes the fluid from the blood and into the brain. So basically burn away this tissue. And they do this, especially in the third world countries where they might not have access to shunt placement, but they even do it in the US as well, actually in quite large extents, mainly on the pediatric population, without actually knowing what it even does to these people. It's pretty crazy, actually. So even though this is life-saving, it's actually quite rudimentary, very invasive, that sort of based on hydromechanical principles along the lines of what a plumber would do to a clogged kitchen sink, where one could either increase the drainage, as you would do with endoscopic third ventriculostomy, or you could lower the amount of fluid coming in by cauterizing the cord plexus. So life-saving, definitely, but the reason why we can't do anything else is that we don't have specific and efficient pharmacological treatment that would reduce this fluid entry into the brain. And that is actually what the patients want. So there was recently a large investigation by the American Hydrocephalus Association, where they asked patients and relatives what should be our highest priority for future hydrocephalus research. And the number one was to develop new treatments that do not require brain surgery to manage hydrocephalus. And the reason why we don't already have this is that we know very little about how fluid actually enters into the brain. And we don't know the molecular mechanism. We cannot target these pharmaceutically to prevent the fluid coming into the brain and therefore reduce the elevated intracranial pressure in that manner. So that's the goal of my laboratory. We want to identify the molecular mechanisms that carry cerebrospinal fluid from the blood and into the brain, or the regulatory mechanisms in order to target these to create the specific and efficient pharmaceutical treatment of at least some types of hydrocephalus or the conditions with elevated intracranial pressure. And this is where Morten's title actually comes in. It's quite useful. This would actually give us opportunities for increased survival and thriving for these patients that otherwise have to go semi-acute neurosurgery for the rest of their life. Genius for Morten. Genius Morten, so impressed. All right, so how is the cerebrospinal fluid secreted? So it has basically been assumed, and I don't know whether you guys remember reading about it in the textbooks, this is probably how much there is about the CSF secretion, basically because we don't know very much. So it's assumed that it happens like how we reabsorb fluid in the kidney collecting duct. That's what the picture is up here. So here you see this monolayer of epithelial cells. That is our kidney collecting duct. It has aquaporins, these water channels that carry water if there is an osmotic gradient, on both sides of the membrane. These are very tightly regulated to ensure our fluid homeostasis is sort of kept up. We have very large osmotic gradients in the kidney. So we basically have an increased osmolarity from the lumen into the cell and an increased osmolarity from the cell and into the interstitium. So we can drive fluid osmotically here. And we are talking several hundred milliosmolar for each of these steps. Very, very high. And I just want to remind you what osmosis is. I'm sure you all know, but generally when people come into a more complex setting, they all seem to forget about it. So just to reiterate, to have osmotic water transport, you need to have two compartments separated by a semipermeable membrane. There must be more osmotic particles in one of the compartments than in the other. And the amount of osmotic particles would determine how much fluid is being moved. And the rate that is being moved in is determined by the water permeability of the semipermeable membrane. So the fluid is secreted by the cora plexus. We have 4 of these in each of our ventricles, and those are also a monolayer of epithelial cells. And here you see a scanning electron microscopy of a rat cora plexus. So you see this very highly invaginated surface area to increase the surface area of the cora plexus. There are aquaporins in the cora plexus. It's the one called aquaporin number 1. And it only resides on the luminal membrane, as you see here with these light gray spheres, that faces the luminal side. There's no aquaporins on the other side. So I would like to convince you that the cerebrospinal fluid secretion, the CSF secretion, does not take place by conventional osmosis as what we see in the kidney collecting duct for 3 different reasons. One of them, you see here, the aquaporin 1, the aquaporin that is expressed in the cora plexus, does not seem to be required for CSF secretion. So here you see a schematic of a cora plexus epithelial cell. You see some abbreviations. The aquaporin 1 is the one in light brown you see here in the luminal membrane. And the only other one I'm going to ask you to remember, you're going to be seeing that many times during the talk, is the blue one, the NKCC1, sodium potassium 2-chloride cotransporter. It's responsible for the chloride gradient in our inhibitory neurons. You might have heard about that during your undergrads. Yeah. So you see on the left side an immunohistochemical picture of the aquaporin 1 being expressed in red in the luminal membrane of the cora plexus. It's sort of this feather-like structure that lies in the brain and waves sort of with the heartbeat that comes along. So humans that don't have aquaporin 1 do perfectly fine. They report no neurological deficits. They have slightly larger diuresis because we also have aquaporin 1 in the proximal tubule of the kidney. But if they are in a scenario where there's plenty of water, they might not even notice they don't have aquaporin 1 until later in life. So they really actually don't give any deficits and nothing's reported neurologically. Same thing for mice. So we knock out aquaporin 1 in, no obvious at least neurological deficiency. They also pee a little bit more and drink a little bit more. So here you see a Western blot showing that there's no aquaporin 1 in the cora plexus of these mice. So even though they don't have aquaporin 1, they of course will actually have reduced water permeability across the cora plexus. So if we take out this tissue, which we could do in rats and mice, of course, and expose them to an osmotic gradient, water would leave the tissue and the volume would become smaller. That's what you see with the black line here. If we don't have any aquaporin 1, it would shrink at a slower rate because water has a harder time leaving when we apply an osmotic gradient. So we've now artificially applied an osmotic gradient. So the water permeability is lower in the semipermeable membrane. But nevertheless, the CSF secretion rate, which you see here in red, is exactly the same in the aquaporin 1 knockout mice. The brain water content is the same, the intracranial pressure is the same, and the ventricular size is the same, suggesting that aquaporin 1 is not required for CSF secretion. Then people would often say, but couldn't that be like some compensatory mechanism? Because these mice are born without aquaporin 1. Could something happen along the way? So we had to address that. So we basically now wanted to knock out aquaporin 1 in the adult mouse. And we do this by a viral approach. So there's a virus we can inject into the ventricles. It's called AAV5, and it generally targets epithelia. And epithelia is this type of barrier cells that the cora plexus consists of. But we have very little other epithelia in our brain. So the epithelia in our brain is cora plexus. So if we inject this virus into the ventricles with a green fluorescent probe, we see it mainly targeting cora plexus. You see here in beautiful green. And we can see again with the Western blot that we see none of this in the hippocampus, that's very nearby, but we see it in the cora plexus, suggesting that this particular virus targets cora plexus. We can then get these mice, aquaporin 1 floxed mice, so they have normal genome, but around the ACAPOR1 gene, they have these LOXP sites. So when we now introduce this Cree element, I'm not going to go into detail, this you probably heard about before, I imagine, into these type of mice, it basically removes part of the ACAPOR1 gene from the core plexus only. And we can then 3 weeks later look at these mice. So 3 weeks later, there is no RNA encoding ACAPOR1 in the core plexus of these mice. There is no more expressed ACAPOR1 on the surface of the core plexus. Very little protein by Western blot. And also, we find that in the kidney, we do not change ACAPOR1 expression. So the whole systemic fluid balance is the same because it stays in the brain. And in these mice, we have exactly the same CSF secretion rate. We have the same brain water content. We have the same ventricular size, basically cementing that ACAPOR1 is not required for CSF secretion. Number two, there had to be an osmotic gradient across an epithelium to drive osmotic water transport, and there is none. So here you see the osmolarity of blood and CSF in rats, in pigs, and in humans. And you can probably appreciate that there is absolutely no difference in osmolarity in these two fluids on each side of the core plexus. And lastly, number three, CSF secretion can occur independently and even against an osmotic gradient. So what you see here is a CSF secretion rate on the y-axis in rats. And on the x-axis, we have the delta osmolarity across the core plexus. So on the right side of the graph, we basically have elevated number of osmotic particles in the ventricles. On the left-hand side of the graph, we have fewer osmotic particles in the ventricles than we do in the blood. And in the middle around the zero, we have the same amount of osmotic particles on each side of the membrane in a live rat. So we can see here where there's 0 milliosmolar difference, which is basically what we found in the rat, we have robust CSF secretion, which is around 7 to 8 microliters per minute. But even more interestingly, on the left-hand side of this graph, now marked in red, we actually have a scenario where we have up towards 50 milliosmolar higher osmolarity in the blood than we do in the ventricles. So water really ought to leave the brain due to osmolarity, and still we have robust CSF secretion coming into the brain. So we can create CSF against an osmotic gradient. And we're not the first ones to have shown this. It's been shown in goats and rabbits and cats back in the 60s and the 70s when they were really, really good at this type of experiments. They've done it with different types of osmolytes. They've changed it in the ventricles. They've changed it in the blood. Everybody always gets the same thing. And this is not exclusive for core plexus. All our epithelia in our body can transport fluid against an osmotic gradient. And this has been known for 100 years. We don't just necessarily know exactly how it happens, so we don't agree on how it happens. Anyway, so with no osmolarity difference across the core plexus, it has then been suggested that there could be a local osmotic compartment in between these microvilli at the surface of the core plexus. And people have tried to find this with ion-sensitive microelectrodes, where you just go closer and closer to the surface to see if there is elevated number of electrolytes there, or osmolytes. They've never been able to find anything. And we did some mathematical modeling, or we didn't do it, but our collaborators did the mathematical modeling, where they basically wanted to ask if it was possible to have a local osmotic gradient between these microvilli that could drive the CSF secretion. And they arrived at the conclusion that less than 0.1% of CSF secretion could be accounted for by this mechanism. So this is where the bumblebee comes in. We know that CSF is secreted, we just simply do not know and understand how. Like back in the 30s, when people first talked about bumblebee flying, where they knew they could fly, they could see them fly. It's just physically, it really ought to not be able to fly. So we would like to actually introduce that this elusive osmotic compartment would reside inside the transport proteins themselves. And this is what we call co-transport of water. So ACAPORs are only able to transport water down the osmotic gradient, and only if there is an osmotic gradient. So co-transporters, you probably remember the sodium coupled glutamate transporters, sodium coupled glucose transporters, these kind of coupled transporters. They've been shown for the last 20, 30 years that they're actually able to, some of them, transport fluid in the direction of the transported substrate in a manner independent on the surrounding osmolarity. And the NKCC1, sodium potassium 2-chloride co-transporter, is one of the ones that have been shown to actually be able to do that. So people crystallized the NKCC1 not long ago. This is human NKCC1, and here you see up towards 100 water molecules lining the path through the transporter. And these, of course, are not going to be stagnant. They're going to move with the conformational changes of the transporter. So very simplicity, it could look something like this. We have a transporter with open binding sites towards one side. You have 1 sodium, 1 potassium, 2 chloride, and some water entering into the transporter and being released on the other side. So basically, each time the transporter turns around, it takes a suitcase of water with it across the membrane. So it's basically a fixed number of water molecules. And I'm not going to show you all the biophysical evidence of that because that probably be a little bit too detailed. But in this type of water transport, it would be the same amount of water molecules that is being moved irrespective of the surrounding osmolarity. So one could create an experimental scenario where we have an osmotic gradient pointing in the opposite direction, and water will flow down osmotic gradients along the pathways there would be, but if the transporter would continue to transport the same amount of fluid in the opposite direction. So co-transport of water. And we wanted to see if this actually took place in the choroid plexus. So here we've extracted the choroid plexus from a rat. We can then add fluid onto the choroid plexus, which has an extra amount of osmolytes. So here we basically add 100 milliosmolar. So normally we have 300 milliosmolar in our blood and in our ventricles, so approximately 300 milliosmolar. So we can add an extra 100 milliosmolar. So it's quite a large osmotic gradient. So we can do this by adding 50 millimolar sodium chloride, would give you 100 milliosmolar. So we do this, and you see a shrinkage of the tissue because water is exiting from the choroid plexus. If we then do this with potassium chloride instead, so it's still an extra 100 milliosmolar, just with potassium chloride, we see exactly the opposite. We see a swelling of the tissue, even though it faces 100 milliosmolar. And we believe this is due to NKCC1 now going inwards with the high potassium concentration. And we believe so because if we add an inhibitor, bumetanide, that inhibits this NKCC1, now the tissue starts behaving like a passive osmometer again. It shrinks when it sees 50 millimolar potassium chloride. So the NKCC1 is able to transport fluid against an osmotic gradient in the choroid plexus. And by doing so, it can actually contribute to CSF secretion along with its 2 neighboring proteins, which we're not going to be talking very much about, so you don't have to remember them. But one is the sodium potassium ATPase, I'm sure you know that one, NKA, and there's also a bicarbonate transporter, NBCE2. Yeah.
So just a quick one, the KCI with the inhibitor, why does it go down instead of just going straight?
Because we still add 100 milliosmolar on the outside, yeah, as potassium chloride. We just also add the inhibitor. Yeah, exactly. And please interrupt if anything is not clear. So we can basically measure the quantitative contribution of different transporters to CSF secretion in a couple of different ways in vivo. And one of them is this Linacre Pearl machine where we can see down through the entire animal. So we could put in a fluorescent probe in the ventricles, very quickly get the animal into the machine, and we can see this fluorescent probe moving backwards in the ventricular system as a proxy for CSF secretion. We can introduce different inhibitors, which we've done with the 3 transporters you see up here, and we can see a reduction in the flow of the CSF backwards in the system, and basically quantify the contribution of these 3 transporters. And we've done this in a couple of different ways, and it gives approximately the same percentage. So I want to point your attention to the NKCC1. So this is here, the CSF flow is reduced by approximately 40% when we inhibit this transporter, meaning that it's involved in CSF secretion. So we can also do this in the more original system that people did this in, ventricular sternal perfusion assay is going to call. I'm not going to go into detail with it, but here we can, again, with pharmacological blockage of the NKCC1, we can reduce the CSF secretion rate by approximately 40%. And this has been shown in mice and in rats and also in dogs over the years, so quite cementing the importance of this transporter in CSF secretion. And when we inhibit this transporter, we can then actually lower the intracranial pressure in healthy rats in this case. So we can here demonstrate that if we modulate the CSF secretion rate, we can also modulate the intracranial pressure, which is kind of the goal clinically in the end to begin with. So people are always concerned about these pharmacological agents, if they're specific, if they're not specific. So we knocked out NKCC1 also by this viral approach I told you about before. So 3 weeks after introduction of this virus in this NKCC1 flocked mice, we see a removal of the protein. We see no expression of NKCC1 at the surface. And we now see exactly the same reduction in the CSF secretion as when we inhibited pharmacologically. And in these mice, we also see a reduced ventricular size and we see a reduced intracranial pressure. So I would say cementing the role. I mean, the reason why we're so detailed with this is because some people really believe that aquaportin-1 has to do all of it. And we have to be sure which transporters actually carrying the CSF, because otherwise we're not targeting the correct one when we want to pharmacologically inhibit it. So now we know that NKCC1 is involved in CSF secretion. So could it actually also be involved in the etiology of some of these pathologies? So post-hemorrhagic hydrocephalus, it's a type of hydrocephalus that occurs secondarily to a hemorrhagic event in the brain. So that could be intraventricular hemorrhage, IVH, or it can be subarachnoid hemorrhage, SAH, or it could be traumatic brain injury, and it can happen with germinal bleeding of neonatals. It happens actually quite often. So it's a pretty high percentage of people that experience some kind of hemorrhagic brain event, and it's very often followed by hydrocephalus. So some of these people, even though the hemorrhage is cleared, they have to have this shunt placed and keep it in for the rest of their lives. And NKCC1 actually is shown to be activated in animal models of intraventricular hemorrhage by us and others that basically leads to an elevated rate of CSF secretion. So now we have CSF hypersecretion in a hemorrhagic event. And it's generally been assumed, mainly by clinicians, that this will be due to an elevated number of osmotic particles in the ventricles with introduction of this blood. But that is actually, and then that osmolarity should be able to pull excess water in. But this is actually not the case. So here we measure the osmolarity in patient CSF. The red ones are the ones that have subarachnoid hemorrhage, and the white one is just controlled. So this is basically people that come in to have removed an aneurysm that hasn't ruptured yet. So there's nothing wrong with them. So we get CSF from these patients as control. So there's no elevated osmolarity. So that's not what drives the extra CSF secretion. What we believe is actually, at least in part, the elevated inflammation that comes along with these hemorrhagic events. So we found 10 different chemokines and interleukins that was elevated in CSF from these patients. And when we take exactly these inflammatory markers and then add them on in the rat version and add them onto a rat core plexus, we can actually mimic this elevated activity of NKCC1 once it sees these inflammatory markers. So in a hemorrhagic event, and with the associated inflammation, our colleagues across the pond has demonstrated this elevated CSF secretion and this contribution to hydrocephalus with an intraventricular hemorrhage model in rats. So they can then inhibit NKCC1 with this bumetanide, and there they actually see that the ventricles no longer get enlarged with the hemorrhage, and we see basically the same size as we do in the control animals. And other group has done with subarachnoid hemorrhage and other type of hemorrhage, where again, bumetanide, so inhibition of NKCC1, reduce these enlarged ventricles. So could we use NKCC1 as such a pharmacological target that we're so in search of? And then somebody's shaking his head. Exactly, no, we cannot. First of all, because we have to give this pharmaceutical agent into the ventricles themselves. If we give it systemically, it doesn't affect the NKCC1. And once we deliver something intraventricularly, we're already doing invasive neurosurgery. And second of all, if it is in the ventricles, it can also penetrate into the brain parenchyma, where it can target NKCC1 in the neurons, especially the interneurons. It's going to change the chloride concentration in these cells. It's going to change chloride's equilibrium potential. Remember back from your first years. So it's going to change the ability of our neurons to send inhibitory signals. That's not going to be good, I think, for treating these type of patients. So we do not think NKCC1 would be a good option. And again, if we give it systemically, it's going to target the many other places NKCC1 is expressed, also like the kidney. So we'll have diuresis in these patients all the time, which is also not be a good idea. So we're basically interested in things that can modulate NKCC1 or some of the other transporters that perhaps could be more specific to the core plexus, and ideally could be targeted from the systemic side. So we don't have to do any kind of neurosurgery to give it to this patient. So we have many different angles in my laboratory focusing on this. And the one I want to tell you a little bit about is this ion channel called TRPV4. It's expressed very highly in the core plexus. It's actually the place where it's expressed the highest in the brain by far. And it's like the second highest expressed ion channel in the core plexus. Got tons of it. So it's a mechanosensitive, lipid sensitive also ion channel. It's a calcium channel. So when it gets activated, we have demonstrated it can activate this kinase called SPAC. And this kinase can actually phosphorylate NKCC1 and make it turn around faster. So we've demonstrated that we can give a TRPV4 activator into the ventricles. We can see an increased rate of CSF secretion, as here shown in green. We can give an inhibitor into the ventricles. This is all done in rats. And we can see a reduction of the CSF secretion. And the latter can also reduce the intracranial pressure, here shown in healthy rats. So TRPV4 could basically be the central conductor of our CSF secretion rate. And we're then interested if that could be our pharmacological target. But this particular inhibitor and activator I show you here, we've given directly into the ventricles. And again, that will not be beneficial for treatment of patients because, again, it is already invasive. So we're interested in finding some kind of pharmacological inhibitor of TRPV4 that we can give from the vascular side. And we tested a few of the published inhibitors of TRPV4. We gave it IP, so intraperitoneally, into these rats, and then measured the brain water content later. And one of these, and it's just called XX here, we're just writing up the manuscript right now, can actually give us a reduced brain water content in healthy rats. And we know that at least in part, this is due to a reduction of the CSF secretion rate, as shown down here in the gray dots. And we've also demonstrated that TRPV4 inhibitor directly inhibits the NKCC1 and the sodium potassium ATPase in some radioactive transport assays. So we can modulate the transporters in the core plexus by giving a TRPV4 inhibitor to the blood side of these animals. So to test this in an animal model of ventricular hemorrhage, we basically inject the rat's own blood into the ventricles. And we can then see this enlargement of the ventricles here shown in yellow and in green. And we can quantify the size of the ventricles and also measure the brain water content. So in this intraventricular hemorrhage model, we have excess fluid coming into the brain. So we can give this inhibitor, IP, either at the same time as we give the blood or even 2 hours after, where we can actually mimic the clinical scenario where a patient will come in after having a hemorrhage and give a medication 2 hours later. And in this setting, we actually see a reduction of the ventricular size, and we can also quantify that it basically approaches that of the control situation, and the brain water content does as well. So we can give this inhibitor to 2 hours after we have the hemorrhage event, and we can then actually reduce the hydrocephalus formation. Yeah.
How long does that last?
We actually don't know. We only measure 24 hours later. So this is 24 hours after they have the hemorrhage.
Right.
Yeah.
Did you say intraperitoneal?
Yeah.
So how deep would that go?
It just basically goes into the peritoneal cavity. Okay. And it normally spreads into the blood vessel almost at the same time as you give it IV. So in humans, you would give it IV and not IP. It's just a very normal way to give to rats. But it would give...
Complicated. Maybe.
No. You're not going to do this in humans. It's just much, much easier than giving IV. And so we basically think that TRPV4 could be this promising target that we've been looking for, especially also because the TRPV4 knockout mice do perfectly fine. They have no particular phenotypes, a little bit of problem emptying the bladder, that's it. So one could actually inhibit TRPV4 over time in these kind of scenarios. So basically, in conclusion, you've done so well with all this molecular stuff. Very good, you guys. So basically, we do not think the cerebrospinal fluid secretion takes place with conventional osmosis. And therefore also that acapone-1, the only water channel expressed in the core plexus, is not important for the CSF secretion and therefore is not a promising pharmaceutical target. We think instead that these 3 transporters, with NKCC1 being the star, contribute to the CSF secretion, and NKCC1 at least by its ability to transport water in the direction of its transported substrates. So in a hemorrhagic model, we can see a hyperactivation of NKCC1, which actually is part of the contributing factor of this intraventricular or the ventriculogamy that signifies the hydrocephalus. And we believe that NKCC1 is important in this because we can inhibit NKCC1 and therefore reduce the size of the ventricles, so basically prevent hydrocephalus in this animal model. NKCC1 is not a good pharmaceutical target because of the anticipated side effects of getting in and targeting the neurons, but bumetanide would be able to actually target as well. But TRPV4 could be such a promising actor that we could go in and modulate this particular transporter, which is also able to activate NKCC1 and able to contribute to this hydrocephalus formation. So we can inhibit this transporter from the blood vessel side, so we don't have to actually do any invasive neurosurgery to do this, and therefore lower the ventricular size and prevent hydrocephalus. So we're just in the very first steps towards our goal of reducing intracranial pressure by a pharmaceutical agent, which we certainly hope will be possible one day, at least for some types of hydrocephalus. We probably can't do all the ones where there's complete blockage of the outflow routes, but in some types of diseases with elevated intracranial pressure. So we are getting closer to addressing this challenge to get an opportunity for excess survival and thriving. What a fantastic title. So that, of course, I'd like to thank all the people that contribute to these many experiments, all the generous funding along the way. And thank you guys for listening.
Wow. Fantastic. This was incredible. I'm sure there are lots of questions. Anybody here? I think I have a question from the back.
So if the NKCC code transports the water, but also transports other things, do you mess with the transport of the other things when you target?
So when you say other things, I mean sodium, potassium, and chloride.
Yeah, the ions, yeah.
Yeah, so you can never transport the water through NKCC1 without also transporting the ions, because it's part of the translocation pathway. So NKCC1 is a good one to target in that setting because it transports sodium and chloride and potassium, of course, into the ventricles, and then potassium is transported back again by the sodium potassium ATPase. So water always follows sodium and chloride. So ideally, if you can only then actually change the sodium and chloride concentration, you're going to change the potassium a bit, but the pump generally adjusts very much of the potassium. You're only going to change sodium and chloride. Whereas if you targeted the NBCE2, the bicarbonate transport on the side, you're going to change the pH of the ventricles. And if you target the sodium potassium ATPase, you're going to change the potassium concentration. It's going to affect the excitability. So you'd be one of the cleaner ways, if you want, to actually target the fluid transport by inhibiting something like NKCC1.
But the sodium and chloride are still altered by your...
Yeah, you have less coming in, but you also have less water coming in. So the concentration of sodium chloride probably won't change, but you will change the amount. I mean, you cannot avoid, you can never only take the water because then you're going to have hyperosmolarity that will pull the water in anyways. So they have to go together.
That's great. Okay.
Thank you.
There's also one there. There's also, if online you have some questions, just let us know.
Just kind of a follow-up from that, so you're with higher concentrations than of sodium and potassium and chloride inside and then the ventricles. Do you also see increased activity in the transporters that bring these ion concentrations or these ions back into or away from the ventricular side and into the lumen?
It's basically if you change the ion concentrations in the ventricles, you can change the activity of all of the transporters.
I
mean, that's always a risk when you fiddle with something, just the alternative might be worse, right? But the sodium potassium ATPase is very well established on this side of the core plexus and also on the blood-brain barrier. It also sits on the side facing the brain. So that can really adjust the potassium concentrations, those two together. Yeah, so the brain is very, very known for if you change the potassium concentration out in the brain or in the blood, it stays very, very constant in the brain, mainly because of sodium potassium ATPase. So it's a type of it comes in different flavors, right? The ones that sits in these particular membranes are very responsive to the potassium. So we've actually seen if you change the potassium concentration, you're going to change the transportivity of both of these two transporters. But together, because one goes up and one goes down when you change the potassium. So you basically keep a constant CSF secretion rate, even if you actually change them because they match each other out. So we actually measured that. It's actually amazing. So we can actually adjust the potassium concentration in the ventricles by these two transporters by keeping the CSF secretion rate constant.
Such beautiful research. This was such a good talk.
Thank you.
There's a question from online. It's a question, but I think I can read it.
Okay.
So it's from Joanna. And she says, thank you so much for the very clear talk. Could you elaborate on TRPV4 being mechanosensitive? I'm interested in the influence of CSF motion, and I'm wondering whether it triggers those transporters.
This is a very good question. So TRPV4 is known as a volume-sensitive channel, so it's known under the umbrella of being mechanosensitive. But actually quite little is known about it, exactly how that happens physiologically. So normally when people put very large osmotic gradients on and they see a large swelling of the cells and then see an activation of these channels. So we actually wanted some experiments where we got the cells to swell slower, so more physiologically relevant, and that does not activate the TRPV4. It's only these very abrupt swellings that people generally do in a very experimental setting. So that particular thing will not happen. So whether it gets activated by an elevated intracranial pressure, nobody knows because it'd be very hard to measure. It'd be very interesting to know actually, because if it did, ideally it should be inhibited by it, but actually it would make it worse. If you have an elevated intracranial pressure and you activate TRPV4, you would secrete more CSF. But whether it could be directly activated or inactivated by that would be a very interesting. We were about to get the knockout mice, so we should be able to address that actually. But it turns out that this channel actually is also activated by synthetic lipids. And we've actually found a natural lipid that comes in with this hemorrhage, LPA, lysophosphatidic acid, that can actually directly activate TRPV4, and you get this activation and get this hydrocephalus from that. So these lipids that are endogenous are not really mapped very well for the TRPV4. So it could be also, it could actually be more of a lipid sensitive channel instead of a volume sensitive channel, even though that's what it's been experimenting with, and with different agonists. So it's we would need to do more work to try to find out how that's actually regulated physiologically or pathophysiologically. So great question.
So Joanna, who is currently in fourth
Thank you.
her son's birthday, 5 years.
Congratulations. Congratulations.
She would often love to talk more to you about it. She's the one who's doing all these beautiful MRI experiments in rats and in mice. And this, of course, is hence why she's interested and she wants to, as I told you, look at how, as if Joanna was here, she would be doing a hand movement like this. As we all love and know. So I'm sure there'll be some interesting thing there. I thought I'd just move it to the human part, because just before doing the seminar, we talked about the potential of actually setting up a trial and thinking about what might happen to young ladies like this one once you put in a shot. And in fact, the other thing that can happen sometimes is you get memory problems when you.
Oh, I have memory problems. I can testify to that.
Yeah, but the beautiful thing is not only your gait will get better once you have a shot, if I'm correct, but also your memory. So there's a real kind of urgency also in order to maybe think about seeing a neurologist and have something done.
I have a son who studied the brain quite a few years ago now at Manchester. And of course, he's very much wanting me to be more supported by. So what you're saying.
Well, we know a few neurosurgeons, so I'm sure we can help you with this kind of thing. But more importantly, not more importantly, but equally importantly, there's a potential study here, right? I mean, one could potentially look at young ladies like you.
Be experimented
and potentially even because,
I'm in my 80s, maybe I'm too old, I don't know.
Not at all, not at all.
I think one of the really cool thing would be, and I'm looking at you, Lisa, would be to use the kinds of tests that Elisa is using, where you hear a piece of music and then you're recognizing whether you've heard that particular part or not, and see whether that could in fact be one of those biobehavioral biomarkers really that could tell us about what happens to your memory very precisely. So I think there could be a basis for something really exciting. As they say at the end of Casablanca. As they say at the end of Casablanca, you know, I think this could be the beginning of a beautiful friendship.
All right, and I can even laugh at that, because the thought of being experimented on perhaps wouldn't be everyone's cup of tea, but I'm up for it.
Yes, milk in before, after. Yes. Any more questions, or should we go straight to the coffee and chocolate? Because she hasn't had enough by the looks of it.
I'm gonna roll home.
They will, of course, also, for those of you who haven't been part of this dance before, not only will we have this, there will be more discussions. You can ask all the questions that you're afraid to ask in plenum, but there will also be Lebanese food. I can highly recommend Lebanese food. Those of you who tried it before, you'll know that it's a joy. Survival and thriving in one package. In food. Should we thank again?